So let's start off with intermediate inheritance, or you may have come across this in the past as incomplete dominance. So, what we're looking at here is a situation where you have sort of a quote unquote dominant allele, that protein is being expressed, but unlike Mendel's expression of a traditional Mendelian dominant allele, the expression of that dominant protein isn't quite dominant enough. So when we get into a situation where we've got a heterozygote, because it's got both copies of that information, it's producing two different proteins in the same cell. That dominant isn't quite strong enough in the way that it gets expressed, and so you get this intermediate phenotype in between. So if I think about this, a lot of times you'll come across expressions, and actually I'll come back to this slide. This is a great example, and this is sort of the classic one that gets used all the time, where we look at things like snapdragons or 5 o'clock flowers are another one that does the same thing, where I take my parent generation, so my purebred generation, here's my purebred red, my purebred white, right? Cross them together, I get that heterozygote in that F1 generation. If this were traditional Mendelian genetics, we would expect that F1 generation's flower color to be red. And yet, instead, we're seeing a third phenotype here. We're seeing that heterozygote looks like a cross between the red and the white. The point I want to make here on this slide is that when we talked about the way that Mendel was coming up with all of this information at the time, there were two big hypotheses going on. There was this idea of a particulate hypothesis, which is ultimately what Mendel supported, the fact that these alleles are discrete traits, they're discrete particles in a cell, and they get inherited separately. The other popular hypothesis at the time, though, was this blending idea, where we look at the characteristics of an offspring and they appear to be some combination of their parents. So if you look at intermediate inheritance, it really does kind of look, at least from our P generation to our F1 generation, like some sort of blending is going on. If I blend red paint with white paint, I get pink. And that's exactly what you're seeing in these intermediate phenotypes when we have an intermediate inheritance pattern. The big difference here, though, is that when we go from F1 to F2, here's yet again why why Mendel's particular hypothesis holds up. Because if this were true blending, my F2 generation would just be a wash of pink, and they're not. All right, so we still get that red back, we still get that white back, but now your heterozygotes look like these pinks. So what I end up seeing is that when we go back to Mendel's original genotypic versus phenotypic ratios, his genotypic ratio from a traditional cross like this, the P to the F1 to the F2, would have been a homozygous dominant, so there's your one, my two heterozygotes to my one homozygous recessive. The difference became when we looked at your phenotypic, you had 3 dominant to 1 recessive. Here in an intermediate inheritance pattern, I still get that 1 to 2 to 1 genotypic ratio, but now it's also my phenotypic. So I have one organism that is homozygous dominant, and in this case shows red. I have one who's homozygous recessive, in this case showing white, and now I still have 2 heterozygotes, but now they have their own unique phenotype. Codominance is another pattern that deviates from that traditional Mendelian genetics. And here, rather than seeing a more traditional dominant versus recessive, both alleles are equally as strong. So there is no masking at all. So when I see Mendel's expression, it's kind of like saying having that dominant allele, having that dominant protein completely masks over any recessive that might be present. Your intermediate inheritance or your incomplete dominance that we just talked about, you maybe get a 50% masking. With codominance, there's no masking at all. They're both just equally expressed. And so if you have both alleles, you have that particular phenotype being expressed. If you've got one, then you're expressing that. If you've got the other, then that's what you're expressing. So we say that they are codominant to each other. So if they're both present, they both get expressed. When we talk about the way in which we utilize the expression of these in writing alleles and writing phenotypes, you'll often see that these are represented by different capital letters in a superscript. Although I will tell you there is some variation in the way people do this. So don't necessarily take that as gospel, but it's often what I'd come across, especially when I was learning this for the first time. But once again, the biggest note here to keep in mind is that codominance is not intermediate. So the heterozygote is not some intermediate in between a dominant and recessive like we just saw with the pink flowers. Rather, they're going to express both traits separately. So the best example that a lot of textbooks will use is talking about your human blood type. So when we look at your blood typing, these are determined by antigens, proteins, markers on your red blood cells. And either you have those antigens or you don't. And those directly then relate to the antibodies that your body produces. So if I take a look, right, it's still a trait that is controlled by a set of homologous chromosomes. So here is dad in this example. He would be what we would kind of consider our traditional heterozygote. He's got an A and an O. All right, as a result, that makes him blood type A. Mom is type B. She's also heterozygous for it. She's got a B and an O. And then we look at what potential children they can produce from that. So dad's able to give his B or his O. So here's O going to this son, B going, or A, excuse me, going to this daughter, O, and O, right? So in this case, if you do the Punnett squares, you can get all 4 children. A child who's type A, here's your true codominant, which is the type AB, a type B, and then a type O as a result. And so you get a lot more variation, a lot more options, if you will, out of a situation where you've got something that's codominant. I will say you can see them expressed like this with it's just the A and the O. You may also come across it like this, where I've got the A superscript and then the I being sort of your traditional type O, just lacking any antigen at all. But here is your true codominant. So because I've got both the A and the B alleles, I express both of those antigens on my red blood cells. Okay, so hopefully intermediate inheritance and codominance are things that you maybe are starting to remember from underclassmen biology. Pleiotropy, however, is probably something that is different and new to you. So here's a situation where I've got a single gene, but when it's expressed, it has multiple phenotypic effects. So having a single defective gene as a result shows up and creates a multitude of problems in your body. PKU disease, cystic fibrosis, sickle cell disease, these are all examples of diseases that would fall into this idea of pleiotropy because it's only a single malfunctioning gene, but the results in which we see disease process and the systems that it affects has a much more far-reaching issue. So sickle cell, for example, the actual misfolding of the protein, we can see the misshapen cell, so there's one potential effect. Then it's not binding oxygen very well, so you have all sorts of issues that go along with that. All right, let's talk epistasis. This one's going to show up a little bit in the eye color case study, and it's actually a really kind of interesting idea. So epistasis occurs where I've got 2 different genes that the expression of one gene at a particular location in your genome then alters the phenotypic expression of a gene somewhere else. And this comes into context with the eye color that we're going to talk about in the case study you'll be doing. So a lot of color expression seems to fall under this idea of epistasis. If I look at the example here in the picture, I'm looking at the fur color of mice. So I've got two genes, one that codes specifically for being either gray or brown, black or brown. Those are your B alleles. But then I also have a second allele that is simply based on whether or not I express color at all. And that color gene, if it's completely recessive, it doesn't matter what the brown versus black allele is. You just won't have any color at all. So if you take a look at this, right? So here I've got two completely heterozygote parents. And here's where these dihybrid crosses really become important. I get alleles that are either two dominant, one recessive for the color of black versus brown, and then a dominant for the color expression. Here's the opposite of that, and then one that's completely recessive. And I see that in both parents. So when we combine these, all of the ones that are completely recessive for that color expression, meaning they can't express color, period, it doesn't matter what the brown versus black was, these guys end up as white. So long as I have one dominant color expression gene, one capital C, then we can start looking at the actual Bs, the browns versus blacks, to see what color they are. So you get this really interestingly unique expression in the way that these work. So if you remember back to dihybrid crosses, if I took two parents who were heterozygous for both traits, I'd expect to see a 9 to 3 to 3 to 1 ratio. 9 who are dominant for both, 3 who are dominant for trait 1, recessive for trait 2, 3 who are recessive for trait 1, dominant for trait 2, and then 1 who is recessive for both. But in the situation of something like epistasis, here I'm actually getting a 9 to 3 to 4 ratio, which is completely different than what I would typically expect from a traditional Mendelian cross. All right, another example of something that steps outside of Mendelian genetics is this idea of polygenic inheritance. And hopefully this is something you had come across as underclassmen as well. So poly means many, right? So when we look at a polygenic trait, we're now looking at a single phenotypic characteristic, but that phenotypic characteristic is controlled by more than one gene. And it can be two, it can be three, it could be four. It really has a big additive effect. The more genes you have that are dominant tends to push you to one end of the spectrum. The more recessives you have, you tend to be on the other. So once again, a lot of coloration works off of phenotypic inheritance, sort of these qualitative characters. And so you end up seeing them in these more bell curve like expression patterns. So here's an example of skin color that happens to be controlled by three different colors or three different genes. If you're completely recessive, you end up way down here on this curve. Completely dominant, you're incredibly dark in the way that your melanin is produced. And most of us are somewhere here in the middle. All right, let's talk about this unique situation. So we talk traditional Mendelian genetics, and we're looking for those sort of 3 to 1 or 1 to 2 to 1 ratios, depending on whether we're talking genotypic or phenotypic. This is an interesting case. So this particular breed of cat, these Manx cats, are bred to be born without tails. They're tailless cats. And if I looked just at the expression at an organism level for this sort of tailless gene, this tailless allele, it's actually dominant to a tailed allele. So as a breeder, you might be going, oh, this should be a great option for me because I get these cats, if they have no tail, that means their offspring should not have a tail unless they end up heterozygous. And so occasionally I'll end up with some of those tailed cats in between. So I would expect if this were a heterozygote mating with a heterozygote, overwhelmingly I should get more tailless cats, right? I should have three tailless to one tailed. Yet here's the actual ratio. For every time we cross two Manx cats, you get two tailless kittens for every one tailed. So instead of a 75% chance of getting a tailed cat, you're getting something more like a 2 to 1 instead of a 3 to 1, right? So my odds have gotten a little worse. And here's the reason. If you start looking at that very first statement, hopefully something jumped out to you. If I do a traditional Punnett square, I should have 4 offspring, not 3. So what's going on here? So in the case of these Manx cats, there's no such thing as homozygous dominant, right? You'd think, all right, I'm going to breed these cats, I'm going to make them purebred tailless because then I can only ever get tailless cats. And yet that's not what you're going to see with this particular breed, and there's a reason for it. It's because having that double dominant allele set, that homozygous dominant, is actually embryonic lethal, so that embryo fails to develop. It's never born. It doesn't exist. There is clearly something that is occurring in that particular gene scenario that prevents the offspring from ever fully developing. Maybe it has something to do with the production of the spinal cord being too short to be a viable offspring. I don't know. But that means that every Manx cat out there has to be heterozygous, which does then once again present some difficulties as a cat breeder. And so these cats tend to be kind of on the expensive side because you will always end up with some tailed kittens, even though you're shooting for that tailless. So it's just something to be considerate of when we're testing these genetic crosses in organisms, because we're looking for these ratios. We're looking for that 3 to 1 or some other ratio we may be expecting. But this could be some potential reasons for why something isn't coming out the way that we expect. There's some suggestion perhaps that this is also maybe tied to certain miscarriages. Clearly more research needs to be done in that area, but it's an interesting thought anyway.